So it's not brand new information. All right, so let's begin. Right away, we actually see some words that we have seen before. These first couple of terms were discussed in our very first lecture of Unit 4, Nervous Tissue Lecture.
So we're skipping this big part to start with this one?
Yes, so you'll finish the brain lecture at home. Watch it
Okay.
at home. Yeah, so it's in your file there somewhere. Yes. Yes, so the brain lecture, you guys will have to watch the rest of that at home. Yeah, so I've given you guys 4 handouts there. Both the lectures, the labeling packet, and the study guide for Unit 5.
So for the pictures that are in the picture that we need to start the exam, so we have to learn the whole names or?
I'll tell you in the videos which ones you need to know.
Oh, okay.
Yeah, so the lecture, it's posted on Canvas. If you watch it, I'll tell you exactly what you need. So if you haven't watched it yet, it sounds like some of you haven't. That's fine. You have until Tuesday, but you'll want to watch it sooner than later so that you know the information and can study it before then. Okay, so let's talk about sensations. Sensation is defined as the conscious or subconscious awareness of changes in our external or internal environment. When we say conscious perception or awareness, that's what perception is. This term is something that we mentioned in that first lecture of Unit 4, conscious awareness and interpretation of sensations. It is primarily a function of the cerebral cortex, which we didn't get to last time. Because we ran out of time, but it's in the video that I have posted. The cerebral cortex is an area of your cerebrum that has these, what we call functional areas, places like the primary visual areas where the sense of sight ends up getting processed. So what you guys are using right now is this area in your brain where the signals are traveling from your eyes into our brain and allowing you to, have conscious awareness and understanding of what you're seeing. You're also using like the primary auditory area because you're understanding and hearing the sounds that I'm creating and able to interpret the words that I'm speaking, which actually is also involving another area of your cerebral cortex called the Wernicke's area. I talk about all that stuff, like I said, in the brain lecture for those of you who haven't watched it. Now, we don't have conscious awareness. We're not mentally aware of every sensation that we are going through at all times. We have subconscious awareness of a lot of things that are taking place internally. We are not like mentally aware when our oxygen levels change in our body, our pH levels change, when our blood pressure goes up and down, that kind of stuff. We might feel the results of stuff like that. We might feel like lightheaded if we're not getting enough oxygen and stuff like that, but we're not, specifically mentally aware when certain sensations are changing inside our body. Now, the nature of the sensation, the type of reaction generated vary according to the ultimate destination of nerve impulses that convey the sensory information to the central nervous system. What that means is, these different sensations and how we react based on them, they all differ in where they essentially end up in the central nervous system, how they get there, which relates once again to that cerebral cortex for those things that we have perception over, for the other sensations that we don't, mentally even know that are taking place. They're taking place and being processed in other areas of the brain. Now, each unique type of sensation, things like touch, pain, vision, or hearing, is called a sensory modality, and we group them into two categories, the generals and the specials. Most of what we'll be discussing in this lecture are the special senses of smell, taste, vision, hearing, and balance. So we'll be talking about our nose, our tongue, our eyes, and our ears. The general sensations, there's actually two types, somatics and viscerals. We'll touch just briefly on some of the somatics, which include the tactile sensations of touch, pressure, vibration, itch, and tickle, the thermals, warm and cold, pain, and then proprioceptive sensations, which involves like our muscles, tendons, and joints. We really don't get into it at all in this class at this point anyway. Anything about visceral sensations of conditions taking place in our internal organs. Remember the term viscera or visceral refers to internal organs, things like your stomach and intestines and that kind of stuff, blood vessels, you know, within the brain and so on and so forth. So we're monitoring things internally like, you know, pressure, whether that's blood pressure, osmotic pressure, whatever, chemical concentrations, you know, calcium levels, oxygen levels, that kind of stuff. Stretch, which could be stretch of the stomach, stretch of the bladder, nausea, hunger, and temperature. Regardless, the process of sensation, doesn't matter what sensation it is, it all begins in what's called a sensory receptor, which a lot of times is just the dendrites of a neuron. In this case, we see a unipolar neuron. We're directly stimulating those dendrites with cold stimulus, which we then convert that energy from the stimulus into the energy of a nerve signal, and we then propagate that signal into the central nervous system so that we can interpret that stimuli acting on within the body, and if necessary, elicit appropriate responses. Like when we're cold, we know we shiver, right? That's a response that we have a way to keep our body warm when it is subject to cold. Now, a lot of times we also have, these things are called capsules. So we have encapsulated nerve endings as well. Encapsulated nerve endings are just where there's basically a wrapping around the dendrites inside, which basically makes them a little bit more sensitive and easier to stimulate. What we also have, and it's only associated with some of our special senses, are what we call separate cells, like in our tongue, where we have these gustatory or taste receptors, which are stimulated by the things that we taste, in this case, sugar. Okay, they are not actually neurons, but they then synapse with neurons that then carry the signal into the central nervous system. So the ultimate result is still the same, a signal that is being propagated into the central nervous system. With these two, it just is more direct. Here we have to utilize a cell before we can then stimulate the neuron. So it doesn't really matter whether it's a specialized cell or just the dendrites of a sensory neuron, the same result is the same signals traveling to the central nervous system. Figures, not important. I just kind of wanted to show you that, you know, what we do as far as sensations goes really is similar when you look across the board at these different examples. We're, you know, just trying to get signals into the central nervous system. Sometimes it's a little more direct than others. All right, let's talk about some of those general sensations. Once again, we're not going to focus on the visceral, so we're going to focus just on the somatics, which includes tactile, thermal, pain, and proprioceptive sensations. Okay, so reading through. Somatic sensations arise from stimulating sensory receptors in places like our skin, where we have a bunch of them. Also places like the subcutaneous layer, which is just deep to the skin. We've also got some somatic sensor receptors in places like the mucous membranes of our mouth, the vagina, the anus. We've also got them in our muscles, tendons, joints, and even within our inner ear. We have, like I said, a whole bunch of them in our skin, and the ones that are in our skin we call the cutaneous sensor receptors, and so those sensations that are arising from our skin are called cutaneous sensations. Remember the word cutaneous, just a fancy word for skin, and we can see several of them here in this picture at the bottom that I'll refer to quite a bit in the lecture. The sensor receptors for somatic sensations are distributed unevenly. In certain areas of our body, we've got a lot more receptors than others, which makes them more sensitive. The places that are the highest density or have the highest density of sensory or somatic sensor receptors include the lips, the tip of our tongue, and our fingertips, which, if you think about it, are the places that come into contact with, stuff the most. When we feel for things and when we touch things and we're trying to, feel textures and that kind of stuff, test temperature, we're using our fingertips. We might put something up against our lips or the tip of our tongue before, we put it all the way in our mouth so that, we don't burn everything in our mouth, just maybe feel a little of that heat right there on our finger or our lips or the tongue, tip of the tongue. So they're going to be just loaded up with sensor receptors. Once again, the 4 modalities or 4 types of somatic sensations are tactile, pain, thermal, and proprioceptive. Tactiles include touch, pressure, vibration, itch, and tickle. And here is some information on the different receptors. There are several types of what we call encapsulated mechanoreceptors. So they look kind of like this. That are attached to large diameter myelinated A fibers, which refers to the axon. We classify axons for these sensor receptors as either A, B, or C. Now this is really kind of physiologically more so stuff than anatomy, but the bigger the diameter, the faster it carries impulses. So A's are the biggest, and then C's are the smallest, so C's would be carrying signals the slowest. We also talked about myelin, like what we see here with all 3 neurons, and how myelin speeds up nerve signals. So these large diameter myelinated A fibers are going to carry signals extremely, extremely fast for sensations of touch, pressure, and vibration. Other tactile sensations like itch and tickle are detected by free nerve endings attached to smaller diameter unmyelinated C fibers. So when we have the sensation of touch, we're carrying that signal in a lot faster than when we are itchy or when we're getting tickled, which seems kind of weird, but that's just the way that our body is built. Receptors involved with touch sensation include what we call corpuscles of touch, or Meissner's corpuscles, as you'll sometimes see them listed as. We find those in the dermal papillae of hairless skin. Remember, dermal papillae are the extensions of the dermis that match with the contours of what we call the epidermal ridges in our epidermis. Here we actually show a corpuscle of touch. Notice there's no hair here. Also in parentheses it says Meissner corpuscle. So going back up, finishing reading it. So each corpuscle is an egg-shaped mass of dendrites enclosed by a capsule of connective tissue. We find them in places like our fingertips, the hands, the eyelids, the tip of the tongue, lips, nipples, the soles, the clitoris in females, and the tip of the penis in men. All places that are extremely sensitive. Hair root plexuses are something we've talked about before. Touch receptors found in hairy skin, always associated with hair because they wrap around the hair follicle and thus make our hair an extension of it. There was a lot of talk last night and this morning online about the presidential debate or vice presidential debate. I don't know if anybody watched it, but apparently there was a fly that sat on top of the head of one of the vice president or the vice president, and it was up there forever. And they said, "What the heck is he wearing a wig?" Because he didn't brush the fly away, and it just sat there forever. But Most people would feel that fly and swat it away. He chose not to, I guess, or didn't even know it was there, more than likely. So, as we've talked about these before, they're free nerve endings wrapped around hair follicles. Merkel or tactile discs, type 1 cutaneous mechanoreceptors, as they're also known as, are also things we've talked about before. Touch receptors in the deepest layer of the epidermis, plentiful in the fingertips, the hands, the lips, and the external genitalia. Those are labeled right there. So some of this stuff we've seen before back when we talked about the skin. Last type of touch receptors are type 2 cutaneous mechanoreceptors, or affinity corpuscles, found in the deep dermis, as well as ligaments and tendons, and are also abundant in our hands and the bottom of our feet, what we call the soles. And they are shown down here in our picture, down deeper in the dermis, whereas the type 1s are right there by the epidermis. So those are all for touch. Pressure involves some of those same ones. Corpuscle touch, once again, Meissner corpuscles, type 1s, and a new one, what we call Pacinian or lamellated corpuscles, which are found in many places of the body, places like the dermis, subcutaneous layer, submucosal tissues underlying our mucous and serous membranes. So like in the nose, for example, inside the mouth, inside of other places. Around joints, tendons, and muscles. The periosteum, remember that's what surrounds our bones. Mammary glands in females, external genitalia, and certain visceral organs. So a lot of places we find these guys, not just the skin like we show it right there. Thermal sensations involve, I skipped something, sorry, didn't I? Vibration involves touch and Pacinian corpuscles. Corpuscles of touch detect lower frequency vibrations, while lamellated corpuscles detect higher frequency vibrations. Itch involves free nerve endings, so do tickle sensations. Free nerve endings are just where the dendrites are uncovered, which is not like what we see over here, where there's that big capsule around them. Thermal sensations involves thermoreceptors, which are also free nerve endings. Two distinct thermal sensations exist: coldness and warmth, detected by two different types of receptors, what we literally call cold receptors and warm receptors. For whatever reason, I've never really researched why, but cold receptors are in the epidermis. Warm receptors are located on the dermis, responding to warmth versus responding to cold. Now, there's kind of a window in which we can stimulate either the cold receptors or the warm receptors, because if you go too cold or too hot, what are you going to stimulate instead of them? Like if you touch something super hot, what's it cause? Pain, right? Okay. So pain is going to stimulate, same thing, free nerve endings, as what we've seen for the last several. The special type of pain receptors are called nociceptors, which I always tell students, remember, most people, if you ask them, do you want to stimulate them? They would say no, because most people don't like pain. We find nociceptors in every tissue of the body except for our brain. So when you have a headache, it's not your brain aching. Stimulated in response to intense thermal, mechanical, or chemical stimuli causing a release of chemicals, ultimately stimulating those nociceptors. And then over on the right is an illustration showing you a portion of, looks like the biceps muscle and its tendon, where we can see some of what we call the proprioceptors, which are the receptors involved with proprioception. Let's talk about proprioception or proprioceptive sensations, to allow us to know where our head and limbs are located, how they're moving, even if we're not looking at them, which enables us to walk, type, or dress even without the use of our eyes. Now, eyes obviously help us tremendously, but we don't need them to do those things. Kinesthesia is a fancy word for the perception of our body movements. So a lot of it relates to like the stretch of the muscle, the tension in the tendon, position of the joints, and the three major types of proprioceptors are muscle spindles, which are these right here, wrap around our muscle fibers and monitor changes in length of the muscle. Tendon organs, sometimes you'll see them listed as Golgi tendon organs, monitor the tension in a muscle, or well, the tendon and the muscle, because the muscles and tendons go together. And then joint kinesthetic organs, which I don't have a picture of, because I don't have a joint. I just have the muscle and tendons are found within and around the articular capsules. Remember, articular capsules connect our major synovial joints, and they monitor the joint position and movement. As far as the images go, I'm not worried about this one, but I do absolutely want you to know this one. Just focus on the all caps portions of the picture. So here where it says, oops, sorry, I didn't mean to highlight that. Free nerve ending. Don't worry about sensus pain, that I'm not going to ask you to label. No type 1 mechanoreceptor or tactile disc, so on and so forth. You know, you don't need to know epidermis, dermis, subcutaneous layer, because in theory, you should already know that. So. All right, that's really all that we will talk about, which, I mean, there's a lot of information, a lot of text on this page, but lumped it all in because at this point, we're really just going to stop talking about those general senses and dive in on our special senses, where we'll talk about the nose first, where we all obviously know the sense of smell takes place, or begins, really. I shouldn't say takes place, because it's not just the nose, it involves neurons that travel into the brain. Just the one blank there to fill in? Okay. Nose contains about 10 to 100 million receptors for the chemical sensation of smell. Fancy word for smell is olfaction. And those receptors are contained within an area of our nose called the olfactory epithelium, which is in the most superior portion of our nasal cavity, covering the inferior surface of the cribriform plate, which is a portion of our ethmoid bone, forming the roof of our nasal cavity. It also extends along the superior nasal concave, which are also components of the ethmoid bone. Looking at our figures here at the bottom, you can see the kind of purple shaded area here in our picture. This is a sagittal section, so we're looking at, looks like the left half of the nose. This white-ish structure coming across the top of the nose is that cribriform plate, the roof of our nasal cavity, and just below that is the olfactory epithelium, as it shows. Okay, looking over here, we're zooming in on this section of the picture. We show a portion of that cribriform plate, and then below that we show the olfactory epithelium that I'll be talking about. Okay. Let's talk about the cells that are found within the olfactory epithelium. The first and very important cells, I shouldn't say they're the most important, because they're all cells are important, but as far as smell goes, these are the ones that are directly involved with it. Okay, these are the first order neurons that transmit the nerve signals from the PNS, from our nose, into the CNS, into the central nervous system. Each of them is a bipolar neuron, which means that the cell body is in the middle of the two extensions. We talked about those in our first lecture of the last unit, which is something you'll need to know for Monday or Tuesday, excuse me. Each of them has an exposed knob-shaped dendrite. On each side of our nose, about 40 bundles of those slender unmyelinate axons of olfactory receptor cells extend through about 20 foramina or holes in the cribriform plate of our ethmoid bone. So looking at the picture over here, we've zoomed in to show that there are these tiny little holes in that cribriform plate to allow the axons of those, and they are the blue cells here in our picture. Here's the cell body in between the two extensions, which is why we call them a bipolar neuron. The axons project out of the nose up into a structure called the olfactory bulb. Okay. So reading through, these 40 or so bundles of axons collectively form the right and left olfactory cranial nerves. Remember, those are the first cranial nerves, which are discussed at the end of this brain lecture that we talked about last time. This is the picture we referred to quite a bit the other day. The fattened end of those olfactory cranial nerves are what we call the olfactory bulbs. And which are paired masses of gray matter at the end of those cranial nerves, number 1. They are located just below the frontal lobes of our cerebrum, which you can see. Here's the frontal lobe. Right there at the tip of that cranial nerve, number 1, is that fattened olfactory bulb. Part of the olfactory receptors that actually respond to inhaled chemicals are called olfactory cilium. Cilium is singular. Cilia would be plural. You also see them listed as hairs, which are just tiny projections on the olfactory receptor dendrites that touch the olfactory epithelium lining, the upper surface of our nasal cavity. And they're where the receptors for the chemicals called odorants, which are just things that have an odor and can stimulate the hairs, are. So looking at the picture, I'll zoom in tremendously here. Here's the singular dendrite, and then down here at the bottom, we show the little squiggly blue lines. Those are the little olfactory hairs, or cilia. Cilium, they were only labeling 1. Those are the things that are stimulated by the chemicals that we smell, what we call odorants. Okay, aside from those blue shaded cells, we've also got some other cells here in the olfactory epithelium, which include supporting cells, which is going to do exactly what it sounds like. They are columnar epithelial cells of the mucous membrane lining our nose, providing physical support, nourishment, and electrical insulation for those olfactory receptors. They may also help detoxify chemicals that come into contact with the olfactory epithelium. There's a third set of cells, kind of the darker blue cells here. In the deeper part of the epithelium, we call these basal stem cells, or they're just listed here as basal cells, located between the base of the supporting cells. And these are the cells that undergo cell division, producing new olfactory receptor cells, which are short-lived, only living about a month or so before needing replaced. So these cells that we talked about first start to die off. And these guys that are darker blue, you can start to see they're elongating for this one here and there, will end up becoming a full-fledged olfactory receptor cell. Also located within the olfactory epithelium is a duct from, or are ducts from olfactory or Bowman's glands, which we show right here. As it shows here in parentheses are the glands that produce mucus, which is then carried to the surface of the epithelium by way of ducts. This mucus helps to moisten the epithelial surface, and very importantly helps to dissolve the chemicals, which helps them then come into contact with those hairs so that we can then stimulate those receptors. The gland itself is actually located more so in the connective tissue, but as you can see, its tube, its duct, carries a secretion of mucus through the olfactory epithelium. Okay, so lot to kind of process here. Let me tell you what to focus on in these kind of 2 pictures. So we'll start over here on the left. Just know the following: the olfactory bulb, the olfactory nerve, the olfactory epithelium. This is really the image because where you can really see stuff. So over here on the left, the bracketed area, the olfactory epithelium. On the right at the top, the bulb. Then you can skip on down to the olfactory gland. And then the basal cell, olfactory receptor cell, supporting cell, and then dendrite and cilia as well. Not the best image. I mean, it's kind of tough to tell stuff apart, but there is still some good stuff in there that I want you to know. So make sure you study it. It should be one of those pictures that's in your labeling packet for Unit 5 as well. Okay, let's move on to taste, which, as you'll find out, is actually super similar to the sense of smell. Instead of taking place in our nose, the cells that help us with taste are located in our taste buds, most of which are found in our tongue. Talk about taste buds here in a minute. Let you finish writing here first. All right. Fancy term for taste is gustation, chemical sensation similar to olfaction, smell. Primary tastes can be distinguished: sour, sweet, bitter, salty, and umami, which means meaty or savory. Any other flavor would be just a combination of those 5. Receptors for taste are located in our taste buds. Most of our taste buds are found on the tongue, but some are found in our soft palate, which is towards the back of our throat, or back of our mouth, really. That's the upper portion, the roof of our mouth. The pharynx, which is the fancy term for your throat, and the epiglottis, which is a structure that helps us from choking. Here's what a taste bud looks like. Here's what an actual taste bud looks like, zoomed in 700 times. And we see kind of a similar situation to what we saw in the nose, with 3 important cells: supporting cells, receptor cells, and basal cells. Let's talk about the receptor cells first, called gustatory or taste receptor cells. Banana-shaped, having microvilli, called gustatory or taste microvilli, or you could replace microvilli with hairs. So there's kind of 4 different ways you can say that. You can call them gustatory microvilli, you can call them gustatory hairs, taste microvilli, taste hairs. All mean the same thing. Little tiny extensions off of the cell that are stimulated by chemicals that we taste. Okay. In our picture, a little bit harder to tell the supporting cells from the receptor cells, but the receptor cells are kind of shown here in blue. They have the one, or they're the ones that have the little hairs coming off. Okay. These microvilli project from the gustatory receptor cells to the external surface through what's called a taste pore, an opening in the taste bud shown right there. Okay. Gustatory receptor cells then synapse with the dendrites of first order neurons in the gustatory pathway, which are these neurons here. So something that's a little bit different is that these receptor cells are not actually neurons. And we showed that actually on that first page with this picture. This is what we're showing here and talking about. Just looks a little bit different in this illustration than it does here. But those are cells that ultimately synapse and thus create signals in the actual neurons. Whereas when we smell, we're directly stimulating the neurons, because that's what those olfactory receptor cells are. These are not. Basal cells are also found in our taste buds, stem cells that function to produce new supporting cells, which are the last cells. Supporting cells surround about 50 of the gustatory receptor cells. And these guys actually develop into these guys. So it's kind of cool. Basal cells develop into supporting cells. Supporting cells then develop into the receptor cells. As far as the picture goes, I want you to know the 3 cells: gustatory, supporting, basal cells, and then the microvilli in the taste pore. Not my favorite image by any means, but it is pretty dang accurate. You look over here, we can see, you know, it's pretty close to looking the same. I know it's kind of hard to tell the cells apart over here, but we can see the pores, we can see the little hairs, which is pretty cool. All right. Now, we find most of our taste buds in little tiny elevations on our tongue called lingual papillae. Once again, anytime you see the word lingual, it's going to refer to tongue. Okay, so these are just little tiny bumps that are on the surface of our tongue. And there are 4 types: filiform, foliate, fungiform, and valate, all of which are labeled here in our picture. The first filiform are tiny spikes, except, and it says it right here straight up, have no taste buds, and thus they have no role with the sensation of taste. Instead, they contain tactile receptors, which we just talked about. Tactile receptors back on this page. Tactile receptors for things like touch, pressure, vibration, and tickle. So they're going to play more of a role, not with taste, but for like, the sensation of touch. When you come into contact with food, you can put stuff on your tongue. You feel that. You sense that as touch. You might help. It helps with, feeling like texture and stuff like that with regards to food and beverages that we ingest. They also help increase friction between the tongue and help food move through the oral cavity, are located over the entire surface of a tongue. In cats, I always talk about how cats have kind of a sandpapery tongue. If you've ever been licked by a cat, you know what I'm talking about, because they've got a ton of the filiform papillae all over their tongues. We just don't have it as prominently, so our tongue isn't as sandpapery. Foliate papillae are the next type, located in the small trenches on the lateral margins of the tongue. Most of those actually degenerate in early childhood, so not so much important for the sense of taste as adults, which leaves us these last two that have any taste buds, which includes fungiforms, which are mushroom-shaped. I guess that's probably why they call them fungiform, because fungi, mushrooms are fungi. They're found over the entire surface of our tongue, containing about 5 taste buds each. And then valate, which are going to form an inverted V, which is kind of handy because the letter V begins the word valate. They're found at the rear of the tongue, once again forming an inverted V shape, and they contain up to half of the taste buds in our human tongue, each of which of those contains 100 to 300 taste buds. Looking at this picture over on the right, it shows a valate papillae. We've sectioned into it, and the taste buds are actually kind of down in the depths of it, which is kind of cool. Each of these little blue dots is a taste bud, and then that is why saliva plays a big role with the sense of taste, because it helps dissolve the chemicals that we taste, get it down into those crevices so that we can stimulate the taste hairs. If you ever want to experience and illustrate the importance of saliva with taste, it's something I used to do in a physiology lab, would be to like dry the surface of your tongue, like just hold your mouth open for a minute or two and breathe, let it dry on its own, or take a paper towel, and you can sprinkle some sugar on the surface of your tongue. You'll actually have kind of a hard time tasting that sugar for a little while because the saliva is not as prominent to dissolve that chemical. But I won't talk more about that at this point. As far as this picture goes, this actually is a picture we'll see later on in the semester when we talk more about the tongue. When we talk about the digestive system, the tongue is very involved with chewing and more so swallowing. So we'll talk about the tonsils and the epiglottis and that kind of stuff. For now, just highlight the 4 papilla that are labeled: valate, fungiform, filiform, and foliate. Don't worry about this picture. So just these 2 pictures in the image. Okay, on to our next sensation. We will talk about the eyes, because we're going to get ready to talk about vision. Now, we'll get into the eyeball itself, which is the actual structure that allows us to see. But first, there's a lot going on with our eyes with what we call extrinsic structures or accessory structures of the eye. So we'll spend a couple slides here talking about those first. All right, we'll start with the eyebrows, which we all know what eyebrows are, hopefully, the little hairy structures above our eyes. They provide facial expression, like when you're confused or mad, our eyebrows go up and down, allow us to have those nonverbal communication mechanisms. Eyebrows also help with providing protection for our eyes from sweat, perspiration, as well as other matter. Eyelids, fancy term for eyelids are palpebrae. Palpebrae is plural, palpebra would be one. And they block foreign objects, protect from excess light, obviously help with sleep, and we blink something like thousands of times a day to help moisten our eyes because they act like a windshield wiper to help give us the moisture across the surface of the eyes. Our upper eyelid is more movable than the lower, and it contains in its superior region what we call a levator palpebrae superioris muscle, which helps open our eyelids, or our upper eyelids, I should say. If I go to this next page, we've got a sagittal section. Here is that levator palpebrae superioris muscle. I mentioned that when we talked about muscles, just what was that, last week or maybe the week before. That wasn't one of those muscles that I had listed to know. You know, we had around our eyes the muscle known as your orbicularis oculi. That was one we needed to know. But your orbicularis oculi is not what actually opens your eyelids. It's more for like helping like squint. That levator palpebrae superioris muscle, which is exactly what it sounds like. It tells you what it does. Levate means to bring up. Palpebrae, we just learned, means eyelid. Superioris, because it's superior. The space between your upper and lower eyelids is called a palpebral fissure. Labeled down here in this illustration with this little bracket. Okay, the space between the two. The angles are known as what we call a lateral commissure, which is closer to our temporal bone, or they're kind of closer to our ear side. And then the medial commissure, which is closer to our nose. Okay. The medial commissure is where we got this, where we have this little round bump. This little reddish bump is called the lacrimal caruncle. It's a fun word to say. A small reddish elevation on that medial commissure containing sebaceous, which remember are oil glands, sudoriferous glands or sweat glands. So they create kind of an oily, sweaty mixture. Whitish material that sometimes collects in the medial commissure comes from these glands, and that's what a lot of times we wake up with, kind of crusted to our eyelids. Associated with our eyelids are also what we call tarsal or meibomian glands, which I can't show you in the picture at the bottom because they're way too small. But they secrete a fluid that keeps our eyelids from sticking together, found in what's called the tarsal plate, a thick fold of connective tissue that gives form and support to our eyelids. So the tarsal plate would be where the eyelashes extend off. Let's talk about those eyelashes, which help protect from foreign objects, perspiration, direct rays of sun. At the base of the hair follicles of these eyelashes are sebaceous glands called sebaceous ciliary glands that release a lubricating fluid into the follicles. Anybody that has not experienced a sty is lucky. Very unpleasant, kind of like a pimple-like structure, happens when you get an infection in those glands or one of those glands. So looking at this illustration, we've talked about everything as far as the accessory structures. Here in a couple slides, when we actually talk about the eye, we will talk about things like the pupil, the iris, the conjunctiva, which we'll go ahead and highlight in this image everything, even though we haven't talked about the eyeball yet. This image over on the right is going to show you the structure that make up what we call the lacrimal apparatus, which includes the lacrimal gland, which is the fancy term for your tear gland, a series of ducts that we use to drain the tears from our lacrimal glands onto the surface of our eyes. These are called excretory lacrimal ducts, or they're just labeled here as lacrimal ducts. Okay. Tears then pass across the surface of our eye, especially as we blink towards the medial surface of our eye, where there are two holes. Okay, two holes that go to these two ducts here, the superior and the inferior lacrimal canals. Okay. The openings to those are called lacrimal puncta, and actually if you look in a mirror and you kind of open up your eyelid a little bit, you can actually see those. Those are what lead to the lacrimal canals, which then lead to the structure called the lacrimal sac, which is found within the lacrimal fossa of our lacrimal bone. We saw that here a few weeks ago. The lacrimal sac holds those tears and then dumps them into the nose through what's called the nasolacrimal duct. Here it's listed as nasal lacrimal duct. Here I have it kind of combined. Nasolacrimal duct is what allows us, especially when we produce a lot of tears, when we cry, to wash that fluid through the nose as well, which is why our nose runs when we cry a whole bunch. And like I said, that is a washing mechanism to kind of just clear everything out. Lacrimal fluid, or tears as we most commonly call it, contains salts, mucus, lysozyme. Its most prominent component though is water, but does all play a role. Lysozyme actually kills bacteria, which is pretty cool. So make sure you know the stuff in this image that's already molded for you. Also, might not be a bad idea to be able to kind of describe, the pun word here, I guess I'll say, the flow chart, how in the direct order that these tears are going to flow, starting with the glands all the way up into the nasal cavity. So be able to, go in the correct order for that. Okay, one more slide here on accessory structures before we'll dive in on the eyeball itself. Let's talk about the muscles that help move our eyes. All right, let's talk about what we call extrinsic eye muscles. There are 6 of them responsible for moving our eyes in almost any direction and provide the finest motor control in the body, which means they are the most precisely controlled. We can directly, it is absolutely fine in how perfectly we can control our eyes. There are 4 rectus muscles. Rectus, we learned in our lecture on muscles, means straight. Oblique means they run at an angle. So the rectus muscles run straight down the eyeball. And when they contract, they pull the muscle in the direction in which they're located. On top of your eye is, I'll zoom in here at it, the superior rectus muscle that runs and connects to the top of the eye. And when it contracts, it allows you to look up. Lateral rectus is in the lateral side of the eye and allows you to look to the side. Okay, this is a lateral view where we've removed the kind of, looks like the right side of our eye. So it'd be like the right half of the face here to show into the eye socket, into the orbit. We can just barely see the medial rectus. It's kind of hard to see because, it's covered by the other muscles. But when you contract the medial rectus, it allows you to look towards your nose. And then the inferior rectus is going to allow you to look down. It runs and connects to the bottom side of the eye, which we show all those muscles and how they contract and where they'll cause the movement to go. Now, notice they all connect in the same spot posteriorly back in the eye socket in what's called a tendinous ring or common tendinous ring. They also insert in the eyeball into what's called the sclera, which we'll talk about on the next page. The other two muscles are the oblique muscles. The inferior oblique is kind of the simpler of the two. Its connection is to kind of the nasal half of the orbit and then comes across the bottom, inserting on the outside of the eye, on the lateral side. Whereas the superior oblique actually also starts kind of like the superior rectus all the way back here, and it comes up towards the front of the eye. It's not this one, but it's in behind it. It then hooks around and takes a right turn and then connects to the lateral half of our eyeball. It connects, or it hooks around, I should say, this kind of pulley-like structure called a trochlea. Okay, so you can kind of see it. I guess I'll zoom in at this picture, how it comes around and hooks and then comes across. And both the rectus, or excuse me, the oblique muscles help with like rolling your eyes, rotating movements. Okay. So reading through, superior oblique originates posterior near the tendinous ring, then passes anteriorly superior to the medial rectus and ends in a round tendon. Tendon extends to a pulley-like loop of fibrocartilaginous tissue called the trochlea. Finally, the tendon turns and inserts on the posterior lateral aspect of the eyeball. Whereas the inferior oblique originates on the maxilla at the anterior medial aspect of the floor of the orbit, passes posteriorly and laterally, and inserts on the posterior lateral aspect of the eyeball. Do not confuse this muscle, which is also something we just recently talked about. That's the levator palpebrae superioris, which I pointed out here on this picture. Helps you bring your eyelid open, your upper eyelid that is, so that we can see when we wake up in the morning or after we close our eyes or whatever. This is a sagittal section, so we're looking at half of the eyeball once it's been cut in half from front to back, where we can see some of those muscles. Really, I only included it because it was in your textbook. It's not really the most important image or anything. I'm not going to worry about this image. This is the image that I want you to know. So highlight the 6 muscles, superior oblique. Don't do the levator, but do the superior rectus, medial rectus. Do the common tendinous ring, the lateral rectus, inferior rectus, and inferior oblique. Actually, at the top, also highlight the trochlea, the little hook, because that's important. Now, the way that our eyes are controlled relates back to, and this lecture we talked about in the video. If you haven't watched it, you'll learn this. But there is literally what we call, where's it at? Number 3, the oculomotor nerves, cranial nerves that control the movement of our eyeballs and the upper eyelid. They are what stimulate our 6 muscles that I just talked about. Now, thinking about the ways that we move our eyes, it's kind of cool. We can contract, for example, both superior rectus muscles, allowing us to look both eyes up. And we can contract both medial rectus muscles, allowing us to go cross-eyed. Can we contract both lateral rectus muscles? No, because we can't look towards opposite sides. Okay, we can contract one medial rectus and another lateral rectus to allow us to look to the left, which both eyes are going to look in that direction. Same thing if we look to the right. Okay, we could contract both inferior rectus muscles at the same time to look down. We can't contract a superior rectus and an inferior rectus, allowing us to look up and down. So it's kind of weird. We can do some together, others we can't. And that's important for controlling only seeing one field of vision, whereas we would see 2 if we looked in opposite directions. So now people that have quote unquote like a lazy eye a lot of times is due to some irregularities that exist in some of those various muscles of the eye. So that's where that kind of comes from. Okay, on to the actual eyeball. Very small structures, but very complicated. A lot going on with your eyeballs. For good reason, they're very important structures. Okay, let's start at the top. Structurally, like I said, it's very small. An adult eyeball is only about 2.5 centimeters, right at 1 inch in diameter. Only about 1/6 of it is exposed, while the remainder is recessed and protected by our orbits, the eye sockets. 3 layers make up the wall of an eyeball. The outer superficial fibrous tunic. The middle layer is the vascular tunic or uvea. And then the inner layer is the retina, which includes a lot of other structures to go with it that I'll get to. Let's talk about the fibrous tunic first. This is, once again, the outer superficial layer. It's actually avascular, meaning it does not receive blood flow, and is divided into 2 parts, the sclera and the cornea. The sclera I mentioned on the previous page is where those muscles that I just talked about insert. And our sclera is what forms the whites of our eyes. Covers the entire surface of our eyeball except where we have the cornea, which is the clear thing that we look through. Let's talk about the sclera first, though, before we get to the cornea. It is a layer of dense connective tissue made up mostly of collagen fibers and fibroblasts, gives shape to our eyeballs, makes it more rigid, protects the inner parts, and as I just said, serves as an attachment site for those 6 extrinsic eye muscles. The cornea is the transparent coat covering the iris and helping to focus light into the retina. Going back to this picture, it was described here under pupil, it says under cornea. So this is a clear membrane that light has to pass through on its way towards the interior parts of our eyes. When a person has like LASIK surgery done, anybody ever watched like on YouTube that procedure being done? It's kind of a cool thing you can watch. They actually kind of cut that and flap it over so that they can then do the laser stuff on the interior parts of the eyes, and they flap it back over. So it sounds kind of gross, but it's kind of cool. The cornea is actually the most frequently transplanted organ. At least that's what I read somewhere when people pass away. And that's one thing that they'll take, clear part of their eyes. All right, the vascular tunic. The term vascular right there is going to tell you that there's going to be blood flow involved. So as soon as the uvea, 3 parts to talk about. The choroid, which is a highly vascular layer behind the retina, providing nutrients to the posterior surface of the retina. It is shown here in our picture. The white is the sclera that we just talked about. And then you have the kind of peach color. Notice the little red and blue dots. Those red and blue dots would be the blood vessels that are definitely prevalent in the choroid. And so the choroid is in the middle, sandwiched between the sclera and the yellow retina that we show here in the picture. Let's talk about the ciliary body, which is the anterior part of the vascular tunic lying between the iris and the choroid. It extends from the ora serrata, which is a jagged anterior margin of the retina, to a point just posterior to the junction of the sclera and the cornea. Ciliary body consists of what's called a ciliary process or processes, protrusions or folds on the internal surface of the ciliary body. Also contains a structure known as the ciliary muscle that changes the shape of our lens, which helps us focus for near or far vision. So looking at the picture, here's the lens. This is a structure that refracts light towards our retina. I'll get to that here in a second. But the ciliary body includes this set of muscles that are shown here in red that have structures that extend off of it that I'll talk about here in just a second, what we call zonular fibers. When these muscles contract, they can change the shape of the lens, which helps us focus for, once again, near or far vision. Okay. Contains muscle. This is accomplished by contraction or relaxation of muscle, changing the tightness of the zonular fibers. Zonular fibers are also known as suspensory ligaments attached to ciliary processes to the lens. Ciliary muscle is a circular band of smooth muscle, meaning that we don't have voluntary control over it. And our ciliary body also secretes fluids that fill the eye that I'll talk about in a minute. Your iris, something that was labeled in that slide a couple slides ago, is the colored portion of our eyeball. So when somebody says they have blue eyes or green eyes, they're talking about the iris. It's suspended between the corneum and the lens and is attached at its outer margin to the ciliary process. The colors come from melanocytes, just like in our skin. Brown eyes have a large amount of melanin, blue eyes very little, green eyes have a moderate amount. A key function of our iris is to regulate the amount of light that enters the eyeball through what's called the pupil. Pupil, I think is something you actually had to fill in. I don't know if I zoomed over for you, sorry. Pupil is the hole in the center of the iris, so that's the black spot. And you can see it's represented here with the brackets. This changes this distance across based on the amount of light that we want to enter our eyes. As you can see over here on the right, I include these three pictures to show what our what our pupil will do. In bright light, we constrict the iris and the pupil gets really small, versus like right now with the lights off in the room. Your eyes will be more dilated so that you're getting more in there, so it's easier to see with what little light there is. We do this because there are two different muscles involved. Okay, circular muscles that cause pupils to constrict, whereas the radial muscles are going to cause the pupils to dilate. Okay, let's talk now finally about the inner layer of the eye where the retina is, where you find 120 million rod-shaped and 6 million cone-shaped photoreceptors. We call these cells rods and cones because of their shape. That's what's shown here in yellow. Now, in this picture, we're looking at a transverse section of the eyeball. So the eyeball's been cut in half, and the top has been brought off. So it's a horizontal cut. Notice that directly straight behind where light is going to go is a little depression. Okay, that's called the fovea centralis. It's really hard and subtle to see. If I zoom in, you can see a little bit better. The fovea centralis is a section of this bracketed off area called the macula lutea. Okay, the macula lutea. I'll zoom out too far in. The macula lutea, or yellow spot, is the direct center of the retina, and the fovea centralis is a depression that contains only cones, lacks coverings, so is therefore the part of the retina that provides the clearest vision. Anybody ever heard the term macular degeneration? It's a common issue where people start to lose, good vision. Macular is for macula. We want as much light hitting here, and that's great because it's going to go straight into it. You start having degeneration there, your vision is going to really suck, especially stuff that's straight on. Okay. Now, a lot of people think that your optic nerve comes directly out straight out the back of the eye. Your optic nerve actually comes out kind of at an angle. The optic nerves are the nerves that carry the sense of vision into our eyes. They're cranial nerves number 2. Just in front of your optic nerves is an area we call the optic disc, which in parentheses you can see is labeled here as the blind spot. Because there are no rods and cones here, any light that hits this area cannot be processed because it's not going to stimulate any cells. And you can actually demonstrate this with kind of a cool little test. You get a piece of paper and you put like a X on it and a dot on it. You can bring it closer to your eye and eventually the dot will disappear. So it's kind of cool. It's kind of hard to do when we're all in different areas. But you could Google it. I imagine you can do it with your phone. It's pretty neat. But we call that the blind spot. It's a natural blind spot because it contains no rods or cones. Within the optic nerve are the central retinal artery and the central retinal vein, which feed and drain blood to and from our retina. You can see those labeled here in our picture. All right, the lens is not a structure that's part of the layers of the eye, but it's suspended from that ciliary body, as we talked about. And behind our pupil, by those zonular fibers, are suspensory ligaments. It is made up of proteins called crystallins, arranged in layers like an onion. I wish we were all in the same place because we could then dissect an eyeball like I do on main campus. I dissect cow eyes, and it's really cool to see the lens when you pop it out. It's kind of like an oblong marble that you can actually cut into and peel off those layers of proteins. Students really get a kick out of that. Just in front of the lens, between it and that cornea, is a chamber called the anterior cavity, which is going to contain a fluid, which, as you can see, is labeled here as aqueous humor. Okay. This fluid, aqueous humor, helps nourish the lens and the cornea. There's actually 2 parts to the anterior cavity, an anterior chamber and a posterior chamber. Both of those have the fluid called aqueous humor. Also associated with this cavity is what we call the sclerovenous sinus or canal of Schlemm, which helps regulate intraocular pressure, so pressure within the eyes. Behind the lens is a much larger space, which is called the vitreous chamber. The vitreous chamber contains a fluid called vitreous body or vitreous humor, which is more of a jelly-like substance, kind of almost like snot. It's really cool. Like I said, I wish we could dissect together and I could show you all these things in real life. A canal exists within that chamber called the hyaloid canal. A narrow channel, inconspicuous in adults, runs through the vitreous body from the optic disc to the posterior aspect of the lens. Okay, a lot going on with the eye. Here's a freebie to you. This image will absolutely be on exam 5. Not everything, but a lot. So let's go through and I'll tell you what to highlight. I'll just start at the bottom here with fovea centralis, and then I'll go through clockwise. So fovea centralis, optic disc, optic nerve, vitreous chamber, hyaloid canal, ciliary process, ciliary muscle, ciliary body, sclerovenous sinus or canal of Schlemm, known both ways. The anterior cavity. Don't worry about the chambers, it's kind of hard, not that important to know apart. Cornea, pupil, iris, lens, zonular fibers, ora serrata, choroid, sclera, macula lutea. And I just remembered I missed something on a slide a couple slides ago when I did not talk about the conjunctiva. So let's backtrack real quick. I completely skipped conjunctiva on this page. So conjunctiva is a transparent mucous membrane covering the inner surface of our eyelids, as well as the anterior surface of the eyeball, which is what was shown on that page. It covers everything under the eyeball except the cornea.
prevents our eyeballs from drying out. The layer that's on the inner surface of the eyelids, called the palpebral conjunctiva, the bulbar conjunctiva, which was what was labeled on that page, covers the surface of the eyeball. It is labeled here in our picture, and it says in parentheses over the sclera. Anybody ever heard of conjunctivitis before? Anybody know what conjunctivitis is best known as? Pink eye. When you get an infection and your eye turns pink due to the conjunctiva getting invaded. So that's our eyes and the structures associated with our eyes. Let's move on to our ears. Something we've discussed before, and the picture at the bottom should look fairly familiar because it wasn't that long ago we talked about the 3 auditory ossicles, malleus, incus, and stapes. The 3 ear bones, hammer, anvil, stirrup, which are found in what we call the middle portion of our ear, middle ear or tympanic cavity. Ears, very similar to eyes, a lot going on with them. And so we will discuss those things here for the next 2 slides. Okay. Our ears are important sense organs, and that's because not only they allow us to hear, they have the sensory cells for hearing, but they also have receptors for sound waves and receptors, or excuse me, they have receptors for equilibrium. So not only hearing, but also balance. 3 major regions make up our ears: the external ear, which collects sound waves, channels them inward. That's what we can see, what we can stick a Q-tip in. Our middle ear, which conveys sound vibrations to what's called the oval window, an opening into our internal ear where the receptors for hearing are located, as well as the receptors for equilibrium. On this page, we really are going to focus more so on the external or outer ear, and then the middle ear, where those auditory ossicles are located. Then on the next page, we'll talk more about these structures that are found in the inner ear, but I'll tell you what to highlight in that picture at the bottom before we do. All right, the parts of your external or outer ear include the auricle, fancy term for your, ear that you get pierced, or you can, scratch or whatever. Another term for it is the pinna. It's the portion of your ear that you can see, supported by elastic cartilage. Kind of 2 major parts to it exist: the helix, which is the rim, and then the lobule, which is your earlobe. So helix and lobule being the lobe that a lot of people have pierced. Ligaments and muscles help attach our auricle to our head. What you stick your Q-tip into is the external auditory or acoustic meatus or canal, a 2.5 centimeter, 1-inch tube leading from the auricle into the temporal bone, ending at the tympanic membrane, which is a fancy word for your eardrum. We talked about this. We actually saw this picture previously when we talked about the bones, but we've talked about Spermus glands all the way back in Unit 1, which are the glands that secrete our cerumen or earwax. Tympanic membrane, as I mentioned, is a fancy term for your eardrum, a thin, semi-transparent partition between the auditory canal and the middle ear, covered by the epidermis outside, lined on the inside by a simple cuboidal epithelium. Between the layers is connective tissue with collagen, elastic fibers, and fibroblasts. We can't see any of that stuff in the illustration, but I promise you they're there. So sound waves cause this to rattle, which then transmits the vibrations to our 3 ear bones found within the middle ear. Fancy term for your middle ear is the tympanic cavity, a small air-filled cavity in the petrous portion of our temporal bone, only 2 to 3 millimeters wide, lies between the eardrum and the oval and round windows, consists of connective tissue layers of epithelium. Okay, the 3 auditory ossicles, once again, are the malleus or hammer, attached to the inner surface of the tympanic membrane, the incus, also known as anvil, attached to the malleus and stapes. Stapes or stirrup attached to the incus and the oval window. Some muscles limit the motion of our eardrum and limit damage from prolonged loud noises. Unfortunately, they're not as effective against sudden sharp noises, which they're so small we don't even show them in the picture. But that's going to include the tensor tympani muscle attaching to the malleus, which increases tension on the eardrum, and a stapedius muscle, which limits the stapes motion and is the smallest skeletal muscle in the body. Several openings exist and are associated with our middle ear. The opening from the external auditory meatus, which is covered with that tympanic membrane. There's an oval window, which you can see is covered by the stapes. The oval window is, along with the round window, an opening to your cochlea. The round window is covered by a secondary tympanic membrane. It's kind of like a second eardrum. And then there's also this tube that runs to our nasopharynx, which is the throat behind our nose. We call this the auditory tube. Also, it's known as a eustachian tube or pharyngotympanic tube. And it helps equalize pressure in our middle ear and explains why if you plug your nose and you act like you're blowing your nose, what happens to your ears? They pop. Okay. This also explains why like chewing gum when you are flying helps from having your ears pop as that pressure changes. Okay, as far as the image goes, everything will be fair game except for, because it's easier just to tell you what not to worry about. So at the bottom, don't worry about elastic cartilage or cerumen. Don't worry about 2 nasopharynx. That's not labeling something, it's just telling where it's going. Don't worry about the internal auditory canal or the temporal bone. I would also label right here where my hand cursor is, label that as what we call the vestibule. A vestibule is spelled V-E-S-T-I-B-U-L-E. Vestibule. I don't know why they don't have the vestibule labeled. It's a very important component of our inner ear. Which is really where all the magic happens, our inner ear, that is, for both hearing and balance. Okay, let's talk about our internal ear. Also known as labyrinth because it is a complicated series of canals. Labyrinth means kind of like a maze. Structurally, there's 2 parts that make it up. The outer bony labyrinth, which is a series of cavities in the petrous portion of our temporal bone. 3 areas exist of the bony labyrinth. The vestibule, which is what I had you label on the previous page, contains receptors for maintaining equilibrium or balance, and is the oval central portion of that bony labyrinth, once again, right in here. would be this general area in this picture. What we're just showing here is the 3 parts here of the bony labyrinth, as well as some stuff over there on the right, which includes the cochlea, the thing that kind of looks like a snail shell, coiled tube arising from the anterior side of the vestibule. This is going to be the organ that contains receptors that respond to vibrations that are created by sound waves, and thus is responsible for hearing. There are 3 fluid-filled chambers, which you can see are opened up and shown here in this picture: scala tympani, cochlear duct, and scala vestibuli. Superior chamber is the vestibule, ends at that oval window we talked about on the previous page. Inferior chambers, this is tympani, ending at the round window. And then the middle chamber is really the most important area, because that's the cochlear duct filled with a fluid called endolymph, where the organ of Corti or spiral organ is located. This is what transduces or converts vibrations into nerve impulses. This is where the hair cells, which are the receptors for hearing, are located, along with supporting cells. At the tip of the hair cells are little microvilli called stereocilia, which change their shape when vibrations occur, and thus are the receptors for vibrations. None of that stuff is visible in the pictures at the bottom. Picture at the bottom. The other part of the bony labyrinth are these 3 what we call semicircular canals, which contain a different type of fluid called perilymph. There's 3 of them. There's an anterior, a posterior, and a lateral. They also contain receptors for equilibrium, and at the end of each of those canals is a swollen enlargement. You can see right there, not labeled, but that's okay, called an ampulla. Inner membranous labyrinth contains a fluid. Once again, we've seen this called endolymph. Contains 2 sacs, which are labeled here, the utricle and the saccule, which are labeled, but this picture at the bottom is not important. On that previous page, one of the things that you had to identify was the vestibulocochlear nerve, the nerve that is cranial nerve number 8 that transmits signals from our inner ear into the brain for balance, coming from this area, which comes by way of the vestibular branch, and then the cochlear branch that connects to the cochlea that carries sound wave or nerve signal, into our to our brain for hearing. As you can see, there's 2 parts to the singular nerve. Vestibular branch goes to the vestibule, cochlear branch that goes to the cochlea. Like I said, this image is not important. I just include it because it kind of zooms in and shows you the chambers, a little bit about the semicircular canals and the vestibule area. But everything that you need to know is in this image. Another one I'm going to tell you right now will be on your exam. I promise you.